This is the presentation on blood. Okay, so first of all, blood's the only type of connective tissue with a liquid matrix we call plasma. And yes, blood is a connective tissue. It transports vital substances, regulates processes for homeostasis, maintains stability of interstitial fluid, distributes heat, and the amount of blood varies with the body size. It changes in fluid concentration, changes in electrolyte concentration, and the amount of adipose tissue. Blood is about 8% of your body weight. The adult blood volume is about 4 to 5 liters in a female and 5 to 6 in a male. Blood cells form mostly in red bone marrow, and we call them formed elements. So you have red blood cells, white blood cells, and platelets. Okay, as you can see with the percentages here, that the formed elements are about 45% of blood and the plasma is 55%. And you see what makes up plasma: electrolytes, water, proteins, waste, nutrients, different gases. Of course, the majority of plasma is water. Types of proteins found in the blood include albumins, globulins, and fibrinogen. And the gases that you're going to find are nitrogen, oxygen, carbon dioxide. And you'll find nutrients, vitamins, and hormones. Now, when we looked at the formed elements, we see we can divide those up. Platelets are about 4.8%, red blood cells, the vast majority at 95.1%, and white blood cells at 0.1%. And then the white blood cells we can further split apart here. We'll get the neutrophils forming most of them, 50 to 70%, eosinophils at 1 to 3%, basophils less than 1%, monocytes 3 to 9%, and lymphocytes at 25 to 33%. Blood draw in the lab is used to detect blood abnormalities, and this is done in the centrifuge blood sample. I expect you've had your blood taken at some point. Plasma, that's the clear straw colored fluid matrix of blood, and as we said, that's about 55% of the blood volume. This is found in the top of the centrifuge tube. So in the centrifuge, we spin the test tube, and heavier materials go to the bottom of that test tube. The red blood cells, they're 35 to 46% in females and 40 to 54% in males. They're found in the bottom of the centrifuge tube. The percentage of red blood cells is what we call the hematocrit. You've probably seen that written on some of the work that's been done on your blood. White blood cells and platelets, they make up less than 1% of the blood volume. And they're found in what we call the Buffy coat, which is in the middle of the centrifuge tube. And this shows that kind of tube after we put it in the centrifuge. And as you can see, the red blood cells and white blood cells go all the way to the bottom. Okay, with the Buffy coat here in the middle, and then the plasma would stay at the top. Whenever you're working with blood, you need to use universal precautions, and I imagine you've heard of this before. It's a set of specific safety measures that are taken by healthcare workers to prevent transmission of bloodborne infectious pathogens in the workplace. It's mainly used for HIV, hepatitis B virus, added hepatitis C virus later, but it applies to others too. Universal means that it is assumed that any patient may have been exposed to bloodborne pathogens. You always make that assumption when you're working with blood. It's estimated that 4 to 7% of new cases of infectious disease are transmitted by unsafe injections, and that's quite a bit. Specific recommendations to prevent infection include use of personal protective equipment like gloves and masks, and the use of fume hoods, sharp containers, and safe workplace practices like hand washing. Okay, let's talk about the formed elements and how they come about. Hematopoiesis is the origin of the formed elements. The formation of blood cells, and the formed elements consist of red blood cells, white blood cells, and the platelets. Blood cells originate in red marrow from hematopoietic stem cells. We also call those hemocytoblasts. Hematopoietic stem cells give rise to more stem cells, and they give rise to more specialized or differentiated cells in response to hematopoietic growth factors. Lymphoid stem cells, they'll give rise to the lymphocytes, and the myeloid stem cells will give rise to all the other types of the formed elements, like the red blood cells and other types of white blood cells and platelets. So this shows you how the stem cells further differentiate into specific blood cells. So let's start with the red blood cells. We also call those erythrocytes. They're biconcave disc shaped. They consist of 1/3 hemoglobin, and this transports oxygen. When oxygen is bound to it, we call it oxyhemoglobin. When it does not have oxygen, deoxyhemoglobin. Now, the red blood cells lack nuclei in mitochondria, and they cannot divide. They can produce ATP, though, through glycolysis. And that's what the red blood cell looks like, biconcave, as it said. A dimple on each side. Red blood cell count. This is the number of red blood cells in a cubic millimeter or microliter of blood. Typically, it ranges between 4,700,000 to 6,100,000 microliters in males, 4,200,000 to 5,400,000 microliters in adult females, and in children, 4,500,000 to 5,100,000. Knowing how many red blood cells are there are useful in diagnosis of certain diseases and to evaluate the progress of certain diseases or of medications. Changes in red blood cell count will reflect changes in the blood's oxygen carrying capacity also. So red blood cell formation specifically is called erythropoiesis. It occurs in the red bone marrow. So low blood oxygen will cause kidneys and liver to release erythropoietin, EPO, and this stimulates red blood cell production. It's regulated by a negative feedback mechanism. And within a few days of EPO secretion, many new red blood cells appear in the blood. Stages in development of the red blood cell include the hemocytoblast, the erythroblast, the reticulocyte, and the erythrocyte. And the average lifespan of a red blood cell is 120 days because they don't have a nucleus and they don't replicate themselves. Nutritional requirements in erythropoiesis: vitamin B12 and folic acid. These are required for DNA synthesis and necessary for the growth and division of all cells. And then iron. This is required for hemoglobin synthesis. Anemia is a condition in which the oxygen carrying capacity of the blood is reduced, and this is due to deficiency of red blood cells or hemoglobin. So this shows you the negative feedback involved with control of red blood cell production. These are the dietary factors that affect red blood cells. And these are types of anemia. Okay, so hemorrhage, bacterial infections or blood transfusion incompatibility, deficiencies of intrinsic factor from the stomach, therefore inadequate vitamin B12 absorption, destruction of bone marrow by different means. These all call decreased red blood cell numbers. In this first case, hemorrhaging, call it a hemorrhagic anemia. Bacterial infections or blood transfusion incompatibilities we will refer to as hemolytic anemia, deficiency of intrinsic factors, pernicious anemia, and destruction of the bone marrow, aplastic anemia. You can also have decreased hemoglobin concentration, and that's due to dietary malnourishment, heavy menstruation, persistent bleeding, ulcers. This results in iron deficiency anemia. As far as abnormal hemoglobin, you have defective genes resulting in abnormal hemoglobin structure. the sickle cell anemia, and defective genes resulting in deficient hemoglobin. Red blood cells are short-lived in this case. This is thalassemia. Okay, and this shows you on the left iron deficiency anemia, and on the right sickle cell disease. Okay, how do we treat sickle cell disease? It's caused by a single DNA base mutation, and it codes for hemoglobin. This results in abnormal hemoglobin structure. Red blood cells are sticky, and they deform into sickle shapes. And then they block narrow blood vessels, and this stops the blood flow to local areas. Oxygen deficiency can lead to more sickling, more blockages, severe pain. And red blood cells die very quickly, resulting in anemia, and this causes extreme fatigue. Now, as far as treatments, children diagnosed at birth receive antibiotics to protect infection in the spleen. Hydroxyurea, given to cause production of fetal hemoglobin, this is more efficient at binding oxygen, will slow down sickling. Transplant of bone marrow or umbilical cord stem cells can cure disease, but has slight risk of disease or death. Experiments to correct mutation in the genome and infuse new stem cells into bone marrow does show promise. Okay, the breakdown of red blood cells. So after months of bending and squeezing through narrow capillaries, red blood cells lose elasticity and they become fragile. Worn out or damaged red blood cells are removed by the spleen or liver. The ruptured red blood cells are phagocytized by macrophages. So then the hemoglobin separates into 4 subunits, globin chains with heme groups. The heme groups break down into iron and biliverdin, and the iron is transported to red bone marrow by the protein transferrin. Biliverdin is then converted into bilirubin, and biliverdin and bilirubin are excreted as bile pigments. Globin chains will decompose then into amino acids. And these are the major events in red blood cell breakdown. So squeezing through capillary damages older the red blood cells. Macrophages in the spleen and liver will phagocytize the damaged red blood cells. Hemoglobin from the red blood cells will decompose into the heme and globin. Heme is decomposed into iron and biliverdin. Iron is made available for reuse in the synthesis of new hemoglobin or stored in the liver as ferritin. Some biliverdin will be converted into bilirubin. Biliverdin and bilirubin are secreted in bile as bile pigments, and then the globin is broken down into amino acids and metabolized by macrophages or released into the plasma. And this shows you that breakdown of hemoglobin. And this is the life cycle of a red blood cell, as we discussed. Okay, white blood cells, also called leukocytes. They protect against disease. Leukocytes have limited lifespans, so they must always be replaced. And they're produced in red bone marrow under control of hormones, interleukins and colony-stimulating factors. 5 types of red blood cells are found in 2 categories. For the granulocytes, we have granular cytoplasm, a very short lifespan. These are the neutrophils, eosinophils, and basophils. The agranulocytes, which do not have noticeable granules, are the lymphocytes and the monocytes. Okay, some of the functions of white blood cells. Diapedesis, this is where the white blood cells can squeeze between the cells of a capillary wall and they leave the blood vessel. Then they migrate towards the infection sites. Leukocytes are directed to the injury sites by proteins called cellular adhesion molecules. In phagocytosis, engulfing and digestion of pathogens occurs. Neutrophils and monocytes are most mobile and active phagocytes. An inflammatory response is also a function. It's a reaction that restricts the spread of infection. It's promoted by basophils, by secretion of heparin and histamine. It involves swelling and increased capillary permeability. And white blood cells are attracted to infection sites by chemicals released by the damaged cells by the process of positive chemotaxis. So this shows you the diapedesis of the white blood cell going out of the capillary. And this shows you what might happen if, say, a splinter went into your skin and some bacteria were introduced. Now, as you can see, the injured cells release histamine, causes blood vessels to dilate, they become leaky, and the neutrophils destroy the bacteria by phagocytosis. And when neutrophils move through the blood vessel walls, migrate towards the bacteria. Oh, I went in the wrong direction, sorry. Of course, neutrophils move through the blood vessel walls, migrate toward the bacteria, and then the neutrophils destroy the bacteria by phagocytosis. I wasn't following the numbering, I apologize. Okay, let's look at different types of white blood cells. Neutrophils. These are granulocytes with small light purple granules in acid-base stain. Lobe nucleuses, there's 2 to 5 sections. They're also called polymorphin nuclear leukocytes, or PMNs. The neutrophils are the first to arrive at the infection site, and they're very strong phagocytes. 54 to 70% of the leukocytes are these, and these are elevated during bacterial infections. Eosinophils, these are granulocytes with coarse granules, and they stain deep red in acid stains. They have bilobe nucleuses. They moderate allergic reactions. They defend against parasitic worm infestations, and this is 1 to 3% of the leukocytes. It's elevated in parasitic worm infestations and allergic reactions. So here we have the neutrophils and the eosinophils. Okay, the basophils. These are granulocytes with large granules that stain deep blue in basic stain. Granules can obscure a view of nucleuses. It releases histamine to stimulate inflammation. It releases heparin to stop blood from clotting. This is less than 1% of the leukocytes, though. It's similar to an eosinophil in size and shape of the nuclei. The monocytes, these are the largest of the white blood cells. They're spherical, kidney-shaped, or oval, or lobe nuclei. They are agranulocytes. They leave the bloodstream to become macrophages. 3 to 9% of your leukocytes are the monocytes. They live for weeks to months, and they phagocytize bacteria, dead cells, and debris. So here we have the basophils and the monocytes. Lymphocytes. They're slightly larger than red blood cells. They are the smallest of the white blood cells. They have a large spherical nucleus that's surrounded by a thin rim of cytoplasm. They are agranulocytes. T cells and B cells are major types of lymphocytes. You can call them T lymphocytes or B lymphocytes. And both are important in immunity. T cells will directly attack pathogens and tumor cells, though. B cells will produce the antibodies. 25 to 33% of the leukocytes are the lymphocytes, and these may live for years. So here we show a lymphocyte. Okay, we can do white blood cell counts. Procedure used to count the number of white blood cells per cubic millimeter of blood. And it should be between 3,500 to 10,500 when we're looking at the count for a cubic millimeter of blood. Leukocytes, we have a high white blood cell count when it goes above the 10,500. Could mean that there's an acute infection, or the person just did vigorous exercise and a great loss of body fluids has occurred. Leukopenia is when you're less than 3,500 count, and this is low. Typhoid fever, flu, measles, mumps, chickenpox, AIDS, polio, and anemia can cause this. Sometimes you may have a differential done for your white blood cell count. This lists the percentages of the different types of leukocytes. The percentages may change in particular diseases. For example, neutrophils increase during bacterial infections, and helper T cells, which are a type of lymphocyte, decrease when there's HIV infection. And this shows these different abnormal white blood cell numbers with some of their reasoning and why it happens. Okay, clinical application, leukemia. So this is a cancer of the white blood cells, and it's classified by the speed of the onset. So it's acute leukemia if the symptoms appear suddenly and progress rapidly. It's chronic if it begins more slowly, and it may be undetected for months or years. It's also classified by type. In lymphoid leukemia, we have a cancer of lymphocytes that are produced in the lymph nodes, and myeloid is cancer of the granulocytes produced in the red bone marrow. Symptoms include excess numbers of white blood cells, fatigue, headache, nosebleeds, fever, respiratory infections, bone pain, increased blood clotting time, which causes bruising and bleeding. Treatments, traditional cancer treatments, chemotherapy, drugs that target the enzymes that are specific for cancer cells, bone marrow or stem cell transplants, and refining diagnoses. Okay, here we have a person with normal blood cells on the left, and on the right we have granulocytic myeloid leukemia. Okay, platelets are also called thrombocytes. These are really cytoplasmic fragments of karyocytes, a type of stem cell in the red bone marrow. It's produced by hemocytoblasts in response to the hormone thrombopoietin. Hemocytoblasts will lead to megakaryocytes, which then lead to thrombocytes. They lack a nucleus, and they're less than half the size of a red blood cell. The normal platelet count is 150,000 to 400,000 per microliter of blood. In thrombocytosis, the platelet count is too high, and it's due to iron deficiency or infection. It carries a risk of abnormal blood clot formation. In thrombocytopenia, platelet count below normal occurs. It's often due to cancer treatments and results in bleeding easily and bruising on skin. Okay, help in hemostasis, or the stoppage of bleeding in damaged blood vessels, occurs by sticking to the broken surfaces. And they release serotonin, which causes smooth muscles in the walls of the broken blood vessels to contract. Okay, this is a good table to get to know. 14.5 tells you a little bit about all of the types of blood cells. Okay, let's look at the blood plasma. This is the clear straw-colored part. It's a liquid portion of blood, 55% of the blood volume, 92% is water. It contains organic and inorganic chemicals. It transports nutrients, gases, hormones, and vitamins. And it helps regulate the fluid and electrolyte balance and maintains the pH. Plasma proteins are the most abundant dissolved substances or solutes in the plasma, and they're typically not used as an energy source. Okay, this chart shows you the different types of plasma proteins. So the albumins, that's 60% of what you'll see. They originate from the liver and they help maintain colloid osmotic pressure. Okay, the globulins, 36%, and we have alpha and beta. They're both made from the liver. The alpha globulins transport lipids and fat-soluble vitamins, and the beta globulins transport lipids and fat-soluble vitamins. Okay, the gamma globulins. Lymphatic tissue is the origin here, and it constitutes the antibodies of immunity. Fibrinogen, 4%. These originate in the liver, and they play a key role in blood coagulation. All right, gases and nutrients. The most important blood gases in the blood plasma include oxygen and carbon dioxide. The plasma nutrients include amino acids, simple sugars, nucleotides, and lipids. And lipids include fats or triglycerides, phospholipids, and cholesterol. The non-protein nitrogenous substances include non-protein nitrogenous substances, NPNs, which include molecules that contain nitrogen but are not proteins. In the plasma, the NPNs include urea, which is a product of protein catabolism, about 50% of your NPNs. Uric acid, this is a product of nucleic acid catabolism. Amino acids, which are the product of protein digestion. And creatine, which stores energy in phosphate bonds and regenerates ATP in the muscles. Creatinine is a product of creatine metabolism. And then there's BUN, blood urea nitrogen. This indicates health of the kidneys. High levels indicate kidneys not excreting urea in normal quantities due to some type of impaired renal function. Okay, the plasma electrolytes. Plasma contains ions that are called electrolytes since they ionize in water and they can conduct electricity. They're absorbed from the intestine or released as a byproduct of cellular metabolism. The electrolytes are found in blood plasma, like sodium, potassium, calcium, magnesium, chloride, bicarbonate, phosphate, and sulfate. Sodium and chloride are most abundant electrolytes that you'll find. Hemostasis. This is the stoppage of bleeding. The actions that limit or prevent blood loss include blood vessel or vascular spasm, platelet plug formation, and blood coagulation. Blood vessel spasm and platelet plug formation are the most effective in small blood vessel injuries. All three mechanisms are necessary in cases of larger blood vessel injuries. In vascular spasm, we have a direct stimulus to the vessel walls or the pain receptors, and the platelets release serotonin, which is a vasoconstrictor. Then the smooth muscle in the vessel walls contracts reflexively. Vasoconstriction helps maintain prolonged vascular spasm. In platelet plug formation, we have exposure of platelets to rough surfaces or to collagen of connective tissues. The platelets adhere to the rough surfaces and to each other, which forms a plug. And in blood coagulation, cellular damage and blood contact with foreign surfaces activates the factors that favor coagulation. Blood clots form as a result of a series of reactions, which terminates in the conversion of fibrinogen into fibrin. Okay, look at these in a little more detail. Vascular spasm. This is stimulated by cutting or breaking a small blood vessel. Smooth muscle in the blood vessels contracts rapidly. Slow blood loss very quickly, and ends of vessel may close completely. Okay, triggered by stimulation of the blood vessel wall, pain receptor reflexes. And the response lasts a few minutes, but the effect continues for 30 minutes. So this allows time for a platelet plug to form and blood to coagulate. Serotonin is released from the platelets, and this causes vasoconstriction, which further helps to reduce blood loss. In platelet plug formation, the platelets do not adhere to the smooth surfaces, such as the endothelium on the inside of a normal blood vessel. So platelets do adhere to rough surfaces, such as linings of the injured blood vessels, to form a plug. Specific chemical factors mediate the process, such as von Willebrand factor, a plasma protein which is derived from both platelets and endothelial cells, which helps activate the platelets and bind them to each other and to the wall of a damaged blood vessel. When triggered by exposure to collagen, platelets send out a spiny process to help with platelet adhesion and aggregation. Platelets release serotonin and thromboxane A. A temporary platelet plug can stop bleeding in small vessels, but coagulation is required to stop bleeding in larger vessels. Okay, in blood coagulation, this is the most effective hemostatic mechanism, which occurs within 5 to 15 minutes. They form blood clots in a series of reactions in which each step activates the next one, and this is called a cascade. It's initiated by two different methods, the extrinsic or intrinsic clotting mechanism. Many chemicals are used in coagulation and are called clotting factors. Vitamin K is necessary for the functioning of some of the clotting factors. Coagulation depends on balance between procoagulants and anticoagulants. And the major event is conversion of soluble fibrinogen to insoluble threads of fibrin, which traps the blood cells. Okay, so this picture shows that sequence. You can see how the blood clots are formed by that meshwork. Okay, in extrinsic clotting mechanisms, this is triggered by blood coming in contact with tissues outside of the blood vessels or damaged wall of the blood vessel. Damaged tissue release tissue thromboplastin or factor 3, which is not found in blood. A cascade begins, which involves sequential activation of several clotting factors. Thrombin converts fibrinogen into insoluble fibrin threads. Fibrin threads stick to damaged blood vessel surfaces and trap blood cells and platelets. This mass is a blood clot, and this is an example of a positive feedback mechanism. Once clotting begins, it promotes additional clotting. In intrinsic clotting mechanisms, this can start without tissue damage. It's activated when the blood comes in contact with foreign surfaces, such as collagen and connective tissue, instead of endothelium of the blood vessel wall, and is triggered by Hageman factor 12, which is found inside the blood. As in the extrinsic clotting mechanism, this begins sequential activation of several clotting factors. Starting with formation of factor X, this cascade proceeds and ends the same way as extrinsic mechanisms, with formation of a fibrin mesh and then a blood clot. And this outlines blood coagulation. Okay, and this is a good figure to get to know, showing you both the extrinsic clotting mechanism as well as the intrinsic. Notice they both start out differently, but they end up pretty much the same. Okay, this is a list of the clotting factors that are involved and where they're synthesized. Okay, what happens then with the blood clots after they're no longer needed? So after a blood clot forms, it retracts and pulls the edges of a broken blood vessel together while squeezing the serum from the clot. Serum is the plasma minus fibrinogen and most of the clotting factors. Platelet-derived growth factor stimulates smooth muscle cells and the fibroblasts to repair the damaged blood vessel walls. Fibroblasts invade the clot, which help to form the fibrous for dense connective tissue, which strengthens and seal breaks in the blood vessel occur. Eventually, plasma digests the fibrin threads and dissolves the blood clot. This is fibrinolysis. Okay, different types of abnormal blood clotting. A thrombus, this is an abnormal blood clot that forms in a blood vessel. An embolus, this is a blood clot moving through the blood vessels. Thrombosis, this is a blood clot in a vessel supplying a vital organ like the brain or the heart. Infarctions, this is the death of tissue which have blocked blood vessels due to blood clot formation. Embolisms, this is a blood clot that travels and blocks a blood vessel in an organ, such as the pulmonary embolism in the lungs. And atherosclerosis, this is accumulation of fat in the arterial linings and can sometimes cause abnormal clot formation. This is a common form of thrombosis. And this shows you normal, and this shows you an atherosclerotic artery on the right. Let's talk about the deep vein thrombosis. This is a clot formation due to pooling of stagnant blood, mainly in the femoral or popliteal veins, or the deep veins of the pelvis. We have serious complications like pulmonary embolism, in which blood clot travels through circulation and lodges in the pulmonary blood vessel, which results in the loss of function in affected portions of the lung. It occurs with prolonged periods of immobility, such as airplane flights. Symptoms of DVT include deep muscle pain, cramping, redness, swelling, and dilation of surface veins, called phlebitis. The clot may break off hours or days after formation. Preventive measures include anticoagulant medications, compression, stockings, and exercise during travel. To prevent coagulation, smooth lining of normal blood vessels discourage the accumulation of platelets and clotting factors. Healthy endothelial cells produce prostacyclin, or PGI2, a prostaglandin that prevents platelets from sticking to the walls of blood vessels. As a clot forms, fibrin adsorbs or latches onto the thrombin, and this prevents the clotting reaction from spreading. Antithrombin inactivates additional thrombin by binding to it and blocking its action on fibrinogen. Some cells, such as basophils and mast cells, secrete heparin, which is an anticoagulant. Okay, in table 14.10, you see factors that inhibit blood clot formation, some of which we've already mentioned. And these are some of the inherited disorders of blood. So we've talked about some of these already. Let's talk about blood groups and transfusions. In 1910, identification of the ABO blood antigen gene explained the observed blood type incompatibilities. Blood types are distinguished by proteins, or called antigens, on the surface of the red blood cells, and these can be determined by the proteins or by the genes that encode them. Safe transfusions require knowing the blood types of the donor recipient and cross-matching donor red blood cells with the recipient's serum and checking for agglutination or clumping. Okay, antigens. These are any molecules that evoke an immune response. If the immune system finds a foreign antigen in the body, it produces antibodies against the antigen. Antibodies are proteins in the blood plasma that react against a specific antigen. In an incompatible blood transfusion, the donor red blood cells evoke an immune response in the recipient, and antibodies in the recipient's plasma agglutinate or clump the donor red blood cells. Okay, agglutination, clumping of red blood cells, which occurs when an antibody in the recipient's plasma encounters its specific antigen on the donor's red blood cells. Only a small number of antigens on red blood cell membranes evoke serious transfusion reactions, so those of the ABO and RH groups. Transfusion reactions, anxiety, breathing problems, facial flushing, headaches, severe pain, maybe jaundice are seen. Okay, the ABO blood groups. These are based on and named for the presence or absence of two major antigens on the red blood cell membranes, antigen A or antigen B. Antigen A and B are carbohydrates, and they're attached to glycosides on the cell membrane of the red blood cells. Antibodies are associated with some blood types. In general, people produce antibodies against antigens that are not present on their red blood cell membranes. In transfusion compatibility, we must make sure that antibodies in recipient's plasma do not bind to the donor's red blood cell antigens and agglutinate and destroy the red blood cells. An example, type A blood contains antigen A on the red blood cell membranes and anti-B antibodies in plasma. This person can receive transfusions of red blood cells that contain the B antigen, so types B and AB. Here's a breakdown of the different blood types by different parts of our population. And as you can see, overall, type O is the most common. Okay, this is a good chart to get to know. So if you are a blood type A, you have the A antigen, and therefore you're going to produce an antibody to what you don't have, which is B. So you're going to produce anti-B antibodies. If you're blood type B, you have a B antigen, you're not going to react against yourself, so you're not going to produce anti-Bs, but you will produce anti-As. Okay, and AB. This person has both A and B antigens, and therefore is not going to produce antibodies against A or B, because it would destroy itself. And O-type means neither A nor B antigens, so both anti-A and anti-B antibodies would be formed if an A or B transfusion occurred. So this is a good chart to get to know also. It tells you what kind of antigens each blood type has and which antibodies it has. And this shows agglutination reactions. Okay, type O is called the universal donor because they have no antigens, no A or B. So anybody could accept them because if you don't have A or B antigens going into your system, there's nothing to produce antibodies against. So O red blood cells can be donated to a person with any blood type. Even if a person has anti-A or anti-B antibodies, the target antigens are missing on the donor's red blood cells. Not occur. Often, when given to a person with a different blood type, it's transfused slowly in small quantities. Now, type AB is called a universal recipient. Type AB blood lacks both anti-A and anti-B antibodies because it's not going to produce antibodies to itself, A or B. It could take any type, any blood type. Due to lack of antibodies in the type AB blood plasma donor, red blood cells containing antigen A and or antigen B will not be agglutinated. And this is a good chart telling you preferred blood types of donors for each blood type. Okay, well, what about that plus or negative that comes after the letters? This is the Rh blood group. It's named for the rhesus monkey in which it was first studied. The group includes several Rh antigens or factors, but most important ones is what we call antigen D. Rh positives is presence of antigen D or other Rh antigens on the red blood cell membranes. Rh negative do not have Rh antigens on the red blood cell membranes. Anti-Rh antibodies form only in Rh negative people in response to the presence of red blood cells with Rh antigen. Could result from a transfusion of Rh positive blood. Initial transfusion is not dangerous since it takes several days for anti-Rh antibodies to appear in blood plasma. But once a person has developed anti-Rh antibodies, the person is sensitized to the antigen. Another transfusion of Rh positive blood, though, will agglutinate the donor's red blood cells. In erythroblastosis fetalis, or hemolytic disease of the newborn, this is a serious condition in the Rh positive fetus carried by an Rh negative mother who's been previously sensitized to Rh antigen. It's a rare condition due to medical care. This shows you the preferred and permissible ABO and Rh blood types for transfusions. So in essence, with the Rh, if you are Rh negative, you can only receive Rh negative. But if you're Rh positive, since you already have the Rh, you're not going to form antibodies to Rh that you can receive negative or positive. Okay, this is an image showing Rh incompatibility during childbirth. Okay, and that's the end of that chapter.